Well, good morning, everyone, and it's my privilege to welcome you to the public thesis defense for Andrew Hinton as a PhD candidate in bioinformatics and computational biology. My name is Jeremy Purvis, and I'm the chair of Andrew's thesis committee. And we're going to try to get started on time because these things can tend to take a while sometimes. And we want to give Andrew all the time he needs to share his research and then dedicate some time at the end for both a public opportunity to ask some questions. And then for the final part of the defense, we'll ask everyone except for the thesis committee to leave the Zoom meeting, and we'll have private dissertation defense. And before Andrew gets started, I wanted to ask Peter Muka. I think Peter wanted to say a few words. Peter and Wesley Burks are Andrew's co-advisors. And I believe Peter has a few things he wanted to say in introduction before Andrew gets started. So go ahead, Peter.
Thank you very much, Jeremy. It's my privilege to introduce Andrew for his PhD thesis presentation in the curriculum in bioinformatics and computational biology. Just quick background, Andrew arrived at UNC in 2017. His work that he'll talk about today, and in addition, other work that he won't have time to talk about today, has been supported by the NIGMS T32 training grant in the BCB curriculum and by a prestigious Gilliam Fellowship that he was awarded by the Howard Hughes Medical Institute. I've enormously enjoyed working very closely with Andrew for the past 4 years as a mentor and frankly, just as a collaborator. He's particularly impressive as a bridge builder. He works really hard to develop and expand advanced techniques of machine learning and then importantly apply those to real biomedical problems. I want to highlight that his collaborations have included many other groups, and it's fantastic to see many of them represented here to attend his talk. He's worked closely on gut microbiome connections to food allergies with the Burks lab at UNC, on nasal microbiome associations with smoking and e-cigarette use with the Jaspers lab at UNC, and on urinary metabolomics predictors for COVID severity with the Henderson lab at WashU. Like I said, all of these groups are represented here today. It's fantastic to see you all. In each of these collaborations, Andrew has been a truly essential collaborator and contributor in that he's the one who's been applying his particularly unique expertise on so-called compositional data. Some of that expertise is what he will share with you now.
Awesome, thanks Peter for the awesome introduction. I'm pretty excited to share with you all some details of some of the things I've worked with. It's been really a remarkable opportunity to sort of spend some time here at UNC and studying bioinformatics, particularly as it relates to food allergies and the gut microbiome. So with that, I'm going to go ahead and jump in to my presentation. I'm going to share my screen. All right, so today the talk is titled Supervised Learning Methods for Association Detection, Biomarker Discovery, and Pattern Recognition in Compositional Omics Data. And as Peter said, the so-called compositional omics data is a quite unique problem within this space that requires quite a bit of attention for such a small problem. So I want to first start off with a little inspiration of how I got here. I thought it would be a good opportunity, if you weren't already aware of some of my strange path to UNC, but nevertheless, I thought it would be a cool opportunity to just kind of refresh. So back in 2008, I started out as an apprentice. You know, I was a structural designer. I ended up getting an associate's in mechanical engineering, and I sort of followed a path that I thought was going to be quite enlightening in shipbuilding and engineering. But in 2010, my daughter, who was 18 months, Madison at the time, she was diagnosed with food allergies. She had her first reaction was anaphylactic. And so we had to take her to the emergency department, and we were just hit with all this information of, wow, now we have to avoid seafood and tree nuts and peanuts, and there's this thing called food allergy. And I'm just like, wow, what is going on? I knew at that moment that I wanted to learn and do something relative to that space. And so that really fueled the rest of my career, if you will. So then I re-enrolled and went back to school. I got my undergraduate in mathematics. I studied some graduate work in model and simulation engineering. And I did this all while working full-time, and I had some promotions along the way that I thought was notable, at least to add. So in August of 2017, we made the decision as a family to come and start the PhD full-time at UNC with the idea and the goal of actually trying to benefit others within the food allergy space. And so that'll really provide sort of the motivation for why I chose this path, and particularly why I study so much with the microbiome. And that just brings us here today with the dissertation defense. So the thing when I first started was there was this interesting idea that there was a connection between a microbiome and food allergy. And I'm just showing you just two sort of examples of this where there have been some associations reported such that the composition of the gut microbiome may in fact influence the future development of food allergy. And so with that, I thought, wow, this would be a good opportunity to maybe start to learn more about microbiome. And so this next slide, I'm just going to kind of talk a little bit about that. And so if you're not familiar, I'll kind of go through and explain. If you're already familiar, this will just be a super refresher. But so what the microbiome is ultimately just a collection of genetic material for microbes that live on the human body or in the human body. These could be at various sites. It could be the gut microbiome. We could be referring to the skin microbiome, or in some cases the nasal microbiome. Any place where bacteria can live, you can then have an associated microbiome. In particular, the way that we measure this is done through sort of microbial community profiling. And we use two techniques, either what's called 16S ribosomal RNA amplicon sequencing, and I'll explain that here in a second, and shotgun metagenomic sequencing. These are two of the most popular techniques currently used, and they have quite sort of dramatic differences, but they kind of answer sort of similar questions. And so what this figure here will sort of demonstrate, at least, is that if we have a microbial community, as I mentioned earlier, we could be looking at the gut microbiome. What we want to ultimately do is try to figure out what bacteria, what microbes, what genes are present. And the way that we would do that then would be either using the 16S-based approach or the shotgun metagenomic approach. In both cases, you want to take the sample and extract DNA. However, the 16S approach, you're going to spend more time amplifying the 16S rRNA gene. That gene has been shown to be highly conserved among bacteria. And when we do that, we can then assign those genes that were sequenced to OTUs, and the OTUs ultimately can be mapped using public databases such as Green Genes or Silva to taxa that we actually know. The thing here with 16S, however, limitations are when we take this approach, we don't get the same level of resolution. So we may be able to identify that this genera is present, but we don't necessarily get the species or even strain level representation. And in certain bacteria like E. coli, you can have two strains that have up to 30% differences in their genomes and can perform entirely different functions. And so that is certainly a limitation. But the cool thing with 16S is that it's a pretty cost-effective approach. And then the second approach is the shotgun metagenomic approach. Here, you extract the DNA and you actually sequence fragments of that DNA, and then you realign those and compare them to reference genomes, and you ultimately can then go in and assign and identify which species, and in some situations, which strains are present. The thing that they share in common is ultimately that they give rise to what is called relative abundance data. And and we'll talk about this here in a few moments, but this is where the compositional data comes into play, because these data, because of the technology, are inherently relative. So before I talk more about that, I just kind of want to point out that there's several studies that link or at least report associations. I'm just going to show a couple here where the gut microbiome is actually associated in some ways to human disease, whether or not that's the microbiome is perturbed such that it causes disease, or whether or not the disease is actually causing the perturbation, or what we typically refer to as dysbiosis, or a shift away from normal in a particular sample. So one of the more interesting things is the link between the gut microbiome and colorectal cancer. I've actually had several opportunities to work with various colorectal cancer data sets, and I actually see this as well, where there is a distinct difference in the microbiomes of those who have colorectal cancer and those who don't. So that's a pretty interesting thing, whether that the community is shifting in response to the disease, or whether the disease, that shift occurred prior to. We also see this in inflammatory bowel disease, where the gut microbiome becomes perturbed in certain situations there. there are certain signatures that are sort of predictive, or not necessarily predictive, but that can actually classify the differences between those who may have IBD versus those who are healthy. And in certain situations, you might be able to get the resolution of whether it's ulcerative colitis or Crohn's disease. And a more interesting one that we might not think about, but there have been some reports where the microbiome are associated with the gut-brain axis, such that it can influence whether or not a person has anxiety or depression. But the one that I'm perhaps most interested in is the fact that we're starting to look at the microbiome as a screening tool, or perhaps even a diagnostic tool for things like colorectal cancer. And so given that, if we're going to use the microbiome in a non-invasive way, we want to make sure that our predictions are robust and things like that. So that's a lot of the reason for further investigation into the composition nature of the data. And so as we've mentioned, and you've probably heard this word several times, and hopefully that'll be a takeaway from this talk, was that the relative microbiome data are compositional. And so what does that exactly mean? And how do we get there? So if we think of high throughput sequencing, which is a technology that underlies the 16S ribosomal sequencing or the metagenomic approaches, they produce data that have uninformative total counts per sample. And so, what does that mean? And so what I want to do is show you just a figure here to kind of put that into perspective. If we look off to the left here, we can see samples. This is from it's a toy example from actual data. Where each bar here represents a sample and the total counts, which are the return reads, when we mentioned in the previous slide, where each read is sequenced and we count those, and then we can figure out what the composition is. The total counts here represent all counts that were read for that particular sample. And you can see, just qualitatively at least, that there's a tremendous amount of variation in total counts for each sample. The thing that's important to note, however, is just because, say, this sample has what appears to be the most reads, it doesn't have any additional biological insight. So we can't draw any information from that such that we can say, oh, well, because we observe more reads in sample A, they're more likely to have disease, whatever. And the reason for that is because there's limitations in the sequencing instrument itself. There's variability in the sequencing process, there's variability in the size of the sample. And because of that, we don't extract information. And so what we're forced to do, and this is an admission you make in the process of analyzing these data, is that you take the data from this sort of count space and we move it to what is called relative abundance. And so this relative abundance space, you can see these graphs look qualitatively the same, but they're quite different. Most notably, this is what we call row sum normalization. We've normalized the sample such that now we're not necessarily concerned about total counts, we're concerned about relative amounts of counts to other things within the sample. And that small transition, that small change in thought has tremendous impacts on how we analyze these data. Whereas in this space, if the total counts were informative, we would just use traditional techniques. There would be no need to sort of explore this in a compositional way. We could just use our traditional statistics. However, when we get into this relative space, the statistics actually don't necessarily work. There are a lot of pitfalls when we try to apply things and think of these data as sort of constrained, where really they're normalized and they're relative. And so, and that unit sum constraint, which is what you can notice is 1 here. So every sample, all the total, the relative abundance will sum to 1, and that creates this unit sum constraint. And this unit sum constraint is what's directly considered to be the core sort of condition for compositional data. And so given that, so it turns out that there actually is a pretty solid foundation for compositional data analysis. And it was really designed for sort of low dimensional geochemical data. But how do we actually apply those techniques to this unique microbiome data, which is actually quite sparse in that we have tons of zeros in observation, and high dimensional in that we have many different taxa that are actually observed. And so that brings me to my overview of my thesis project. And so as you can see, that there's a strong focus on microbiome. And so a lot of the work that we've done has been focused on developing techniques to actually sort of transition and adapt traditional compositional data techniques to this really unique microbiome data. And so one of the first methods that we developed is this method we call selection energy permutation. Is actually inspired by a method that was developed here at UNC by Steve Marin's group, which is called direct dipro perm, so direction projection permutation. And in that method, what we do is we sort of look out to detect what we call sparse associations. And what that ultimately means is if we observe, if we do a community profile, we identify 300 taxa, all 300 taxa in general aren't necessarily related. While it's great that we observe all these fancy taxa, it may be that the thing that contributes to the disease or the groups or the separation that we're interested in, it may only be a few taxa that are actually really associated. And then the other question becomes that because these data are so high dimensional, how do we actually do that in a responsible way such that we extract the insight, but we can ensure that we're not like overfitting or we're drawing sort of spurious findings. And so in that method, we've actually applied it. We had a really cool collaboration with the Jaspers lab. Elise and I had an opportunity to work with, well, I had an opportunity to work with Elise, who had actually done this really cool experiment on the nasal microbiome that looked at differences between exposure, such that we have a sort of a non-exposed group, those that don't smoke or using cigarettes, and we have another group that were e-cigarette users and another group that were smokers. And we were interested in trying to figure out if there was an association between the nasal microbiome and the exposure, such that our smokers' nasal microbiome different from that of those who don't smoke. And so we actually have a paper that's currently in revision that we applied the cell energy PERM method to, and we were able to show that there is in fact an association between the nasal microbiome and the exposure to cigarette smoke. And so from that, the thing I should have mentioned as well was that the selection energy permutation method is a biomarker discovery method and is an association detection. And so associations are where we're really trying to figure out if there's some relationship between, say, the microbiome or the composition and a treatment or a group of interest. So we don't make any statements about prediction, so this wouldn't be applicable in sort of a diagnostic. It's more so to establish that there exists at least associations. Whereas with differential compositional variation, we build on some of the ideas that we present in cell PERM, and we look at, again, a biomarker discovery that we look for, particularly for sparse associations, but we focus more here on prediction. And so prediction is that we want to actually then apply these methods in diagnostics. We want to use these methods to get the highest prediction in the most robust fashion. And using that, we've actually applied in this method, we've applied some of the things we learned, and just as I mentioned before, that we have associations between gut microbiome and many diseases. We were able to show in this work here that the gut microbiome is actually associated with food antigens such as sensitization, such that the different proteins that mice in this particular study were exposed to modified or at least appeared to change the microbiome in a sort of predictable way. And then what is a really notable sort of application of the DiCoVarML method here is where we had an opportunity, as Peter mentioned earlier, to work with our collaborators at WashU, Laura Marks, John Robertson, and Jeff Henderson, and as you can see, other folks on the paper there. We had a cool application where we applied the DiCoVar to predict COVID severity from urine metabolomics. We're currently working on this paper, finalizing it. And we've also submitted a provisional patent to patent the biomarkers and the technique that we use because we were able to predict with a reasonable amount of certainty who was going to develop COVID severity from just looking at the metabolomics of their urine. So given that, so I've kind of given you a really large overview of the projects that I've done, or at least had an opportunity to work on while I was here. But I'm going to focus this talk on, because we just don't have enough time to talk about all this in detail. I'm going to focus this talk on specifically the DiCoVar method, which is one that we just recently submitted and is currently in review. But before we start, I got to give you some background. And so I love background. And so I'm going to give you just a little bit of background on compositional data so that you can at least appreciate why we have to treat these data differently. And then ultimately, this is the motivation for us to develop these methods in this level of detail. And in particular, I'll talk about two things: the simplex sample space, which is different from the Euclidean space or the normal multivariate space that we would do traditional statistics in, and then how we can sort of overcome that by way of a log ratio transformation. In particular, this has been one of the really important books. In fact, in compositional data, this is perhaps the most important work that exists because it really put forth at least a lot of these ideas at the early stage. And so with that, so I mentioned that there's a simplex sample space. You may not necessarily be familiar with the simplex sample space. So I'll give you some formal notation. So if we have the simplex sample space, if we have a compositional vector, we can take a D part vector. If we measure some object and we get D parts and we look at that from a relative perspective, we can express the simplex sample space using this notation. The thing here that you can take away is that this is the unit sum constraint on each part. And this is a constraint that requires each part to be positive. And so in order for this data to reside in the simplex in sort of this constrained space and the real Euclidean space, or this constrained subspace within the real Euclidean space, we have to have this condition. There are more relaxed conditions that will not necessarily include zeros. However, here, at least what we're presenting, we do in fact consider zeros, which are actually somewhat of a problem. And the other thing that I should note here, and I don't have notation to show this, however, but this little d is basically d minus 1, and that the simplex space is of dimensionality d minus 1, whereas in real space we'd be talking in d dimensions. And so this is just formal notation, but we can think about this more in a practical sense, where if we take some data, we measure some samples, and this is the red points here represent data that are unconstrained in real space. However, when we make that data relative, when we make the admission that those total counts are uninformative, we then normalize the data, and then that projects these data onto the simplex. So if we were to think of the simplex in the 3D space, now you can think that we've restricted our possibilities. So that the possible places that these data points can reside are just on this triangle for this particular example. And we can further think about that in a ternary diagram, where we can look at these data. And so the thing I want you to take away here is that the way that we can measure on the ternary diagram is simply by looking at this space and saying, okay, well, if it's 50% abundant, 100% abundant, we measure along this line, we measure along this line. And this line, we have our Berry center here, and this location is really the data in the actual simplex. So while we see that these data are 3D, in sort of the simplex, again, this is a two-dimensional simplex, this is a two-dimensional figure, and so that's the sort of constraint that those data are required to live there. However, we can do log ratio transformations, and we can take that data and get it out of the simplex, and we can put it back into real space. The real space here, we're not constrained any longer to the simplex, but we are on a hyperplane, and so there's a sort of a nuance in that. But nevertheless, we can put it in this space, and we can actually start to do more traditional statistics. And so given that there's many log ratio transformations you could choose from, each have their sort of strengths and weaknesses. The most simple of them all is the additive log ratio transformation. If we take that same sort of X compositional vector we referred to earlier, this is essentially the way that we would transform those data into, say, Z here. Graphically, we can think of that as, if we have just 3 parts, we can think of that as ratios in this sort of format, where we have a center that we have to define. So we have to define a reference, and then we have ratios between the other 2 parts. Most notably here, where we have 3 parts, we only have 2 ratios in an additive log ratio. So the additive log ratio space is actually d minus 1 as well. The center log ratio is really great at preserving distances. However, it has some limitations when it comes to sort of subcompositional coherence, in that you'll notice that this transformation takes advantage of a geometric mean. That's what this notation sort of represents. And it's the same log transformation that we see here. It's just that the denominator, the reference term here, is actually pulling information from all data points within the sample. And graphically, we can think of that as 3 parts. So CLR, we get 3 ratios, we get this. And finally, the thing that we found most important, and we believe would enhance sort of biological insight, whereas with ALR, we have to define a reference. With the center log ratio, we lose our ability to sort of generalize beyond just the samples we have. But the pairwise log ratio transformation is a quite independent way of looking at these, but it's computationally more intense and more complex. And you can see even just in the notation how we define this, but pairwise log ratio sort of is this combinatorial problem where for every part, we basically choose 2 parts, and we compute, or we can think of every possible combination. In this particular setup, the way we designed it here, what we'd be interested in is the lower triangular matrix, or sort of lower triangular part of all pairwise of this vector. That's again shown by this notation. But the takeaway here is the dimensionality is of D is D squared. So it's basically quadratic. And so what that means is that for 5 ratios, for 5 parts, we may have 10 ratios. If we go to 50, we have 1200 roughly. But if we double 50 to 100, you would think we double ratios, but we don't because it's quadratic. And we go up to 5,000, and quickly you can see that this gets out of hand. And so then the question then becomes, how do we deal with that? We know pairwise log ratios are really important, but then how do we actually deal with that dimensionality increase? We really don't want to use the ALR. We know that there's some interesting limitations with CLR, and we believe that the pairwise log ratio transformation will give us some added insight. And so that's where the DiCoVar framework comes in, and we specifically look out for ways to actually utilize that more robust but higher, ultra high dimensional pairwise log ratio space. And so I'll spend the rest of the talk talking specifically about the DiCoVar framework. So one of the first things I want to sort of level set here is that early on, and this is really prominent in any compositional data, so the literature, Carl Pearson, who invented the product moment correlation. So if you're familiar with correlations, this is the person who invented that, but he noticed early on that there were some spurious correlations that could give rise when we look at relative data. And that sort of has permeated throughout sciences. And so we're aware that when we take correlations, and it's quite easy to show that when we compute these correlations on relative data, depending on the subset that we use, we can get different correlations for the same parts, depending on what's available in the sample. And so that's probably the most important warning to why you wouldn't use traditional statistical techniques. However, in the field, there is this thought that machine learning, which is this really powerful computer technology that can learn things on its own magically, we just think machine learning will just overcome that. And so what I really want to show in this example is that machine learning is also susceptible to the same things, to the same limitations, if we don't treat the underlying data. And so what we're seeing here, I'm just going to give you a toy example. It's a 3-part example in a ternary diagram, as we saw before. But what I want to note is I create a test and a training partition. So the training partition is where we're going to look at, and we're going to train our models. And ultimately, we want our model to define or to learn this decision boundary, which is we're simulating. We're creating this decision boundary through careful sort of simulation. And we expect a well-trained machine learning model will be able to take this decision boundary, learn it, and then generalize it to data that it hasn't actually seen. And so that's what this example is. This is our test set. And so what we're going to do in microbiome literature and in general, random forest is the go-to model. It's a really powerful, I won't get into specifics, but it's a really powerful classification algorithm. And so when we use the relative abundance, the center log ratio transformation, and the PLR, you can see that on our training set, each model does a great job. We use cross-validation. So this is the estimate you may be tempted to report. However, when we actually take those models that were trained and apply those to our test set, which we didn't train on, however, a really strong model that learned real patterns should be able to generalize those patterns to data it hasn't actually seen. But you'll see that on our test set, we lose all ability, at least for these relative abundance and center log ratio transform data, to discriminate between classes. And this is most notably seen just simply by the coloring here. And so each model for each data point, you can think that there's like 30,000 individual data points shown here, and we color each one of them based on the probability, such that the red color is a higher probability that any sample that resides in this region is a class 1, and blue, would be darker blue would be any sample is class 2. But you can see here where the decision boundary is. The model does great. You can sort of see this blue-red transition, but out in this region, we lose that ability. Same thing here, we do great for the most part, but then we lose the ability to generalize that. However, with the pairwise log ratio transformation, we can actually learn this is a really simple linear boundary here. We can actually learn that boundary and generalize those patterns. And so this limitation should at least give pause to just using relative abundance raw and transformed if we're really going to develop diagnostics to diagnose disease or screening tools and things like that. We need to make sure we're treating these data sort of carefully. And so we know that the pairwise log ratio is more robust. But we have this issue with the dimensionality. And so we get the situation where, in general, when we're doing these microbiome studies, we have N here being samples, P being features, in the case of pairwise log ratios, but we have many more pairwise log ratios than we actually have samples. And so that creates a problem because it becomes quite easy to identify a subset of pairwise log ratios that are predictive of anything in that space. And so we have to be very careful in how we apply that. And so one of the first things we want to do is figure out a way to handle the extreme dimensionality. Like, how do we actually figure out, or how do we use or select a subset if we don't want to actually consider, in some situations, 20,000, 40,000 ratios? And we understand that all of them may not actually even be informative. And if we have enough noise, we can actually hide true signal in that data. So it's super important in these sparse association settings to identify the subset that's really contributing to the phenotype that you may be interested in. And so to do that, the way that we propose, at least in our cell energy perm, which is one of the first projects I mentioned, is we compute, we come up with this idea of a differential compositional variation score. Importantly, this is a supervised filtering method, and we compute these 5 statistics, and they all measure something slightly different, except in the case of these two, where we look at location differences. Here, we're interested in scale differences, distributional, and entropy reduction sort of differences. And so what we're trying to do is take each log ratio and figure out how likely, in some ways, not in a probabilistic way, but we're trying to, from a score way, figure out how much information or will this log ratio provide in separating the 2 classes that we're interested in. So again, being supervised, we know, which parts, which samples and ratios belong to class 1, and we know which parts belong to class 2 in a binary sense. But we want to be able to apply that information and draw. We want to essentially learn. So we have examples and we have labels. So we have the test and we have the answers. And so in this situation, we can take those, we can compute our scores. We z-score normalize this. This is a sort of important step because all of these are on different scales. Then ultimately our score then becomes a measure, basically the sum across all these sort of z-score normalized data. And the thing I sort of want to point out too is that we see, at least in this particular example, that these aren't perfectly correlated in many cases. We do have some lower correlations, which means that we have a diverse set of metrics that are capturing some small difference in those distributions. And so that's what we sort of propose, like, okay, we'll rank these ratios and we'll then choose from the ratios that we think are most likely to contain information that's important. The thing here is this is purely univariate. So we're looking at each ratio individually. We're not actually looking at which ratios will work best together. We'll do that at a different step. But then the problem becomes we have a way to rank these ratios, but then how do we figure out if we want to limit the parts? So we can do feature selection at 2 steps. We can do feature selection at the log ratio level, or we can do it at the part level. But we've already said before that when we look at these relative abundance data, we can't extract information directly from the parts. And so, well, do we go from log ratios to parts? And so what I'm trying to show in this example, this is a data set where we have some groups, and we have, this is just a part matrix for 2,965 parts. There is signal here, but because there's so much noise and we have so many dimensions, you can see that the signal is obscured by just dimensionality. And so how do we, if we wanted to develop a diagnostic and we had a constraint where we only can really use 20 parts in our model, and this is actually motivated by our collaboration with the Jeff Henderson lab, where the motivation was to identify a set number, or a small number of features or parts in this case that are most predictive. That's kind of different from the way we think of prediction. And that's what's unique about DiCoVar, is that while we're interested in getting the highest prediction, we're interested in getting the prediction in such a way that we can develop a diagnostic that's cost effective and useful. And so it gets into this idea of cost versus accuracy trade-off, at least what I'm proposing here, is that as we increase the number of parts, we can increase our model accuracy. But if cost were no option, then obviously we would just simply pick the number of parts that maximizes the accuracy. However, when we get into regions or situations where cost is of interest, and we may be limited by certain things, then maybe we don't want to exceed per diagnostic or per screening test a certain threshold. And then that means, how do we, given that cost threshold, how do we select the optimal set of parts that will maximize accuracy given that? And so that's the other sort of problem that we had to address with the DiCoVar. And the solution that we came up with was working with Peter and being a network expert. We use a network-based multi-level feature selection technique. And the reason I call this multi-level is because we're doing the feature selection at two steps, at two stages. We're looking at the feature selection at a part level, and we're looking at feature selection at a ratio level. So we're kind of getting a 2-for-1 here. And so we're going to go back to sort of those diagrams where I showed earlier. We can represent visually the log ratio space as a network. And so visually this is useful, but we can't extract any insight from it directly. It's great to see that we have a lot of connections and it looks great on the picture. However, how do we actually start to use methods with this data? And so what we thought was, well, we have this differential compositional variation score. We can use that score and rank essentially each one of these edges. I should point out, if I didn't already, that each dot here, circle, represents a node in our network, and each line is an edge, and it represents a ratio. And in this case, it's a log ratio. So basically, we can compute this, what we call a differential compositional variation score for each one of these. And we have, with computational methods, ways to do this quite efficiently. So it's not as scary as it sounds. We can implement this in code in a way that we can compute this in a reasonable amount of time. And so what we do, we can actually, then now we have a network we can work with. We get what it's called a weighted network, where the edges now are weighted based on the differential compositional variation score. And at least in terms of this sort of visualization, the color is our strength directly, and then the size is also a measure of strength. And so you can think of the darker nodes here being more important to separate the classes, and the lighter nodes as being less important. Same thing with the edges. The darker edges are thicker and more likely to contain higher differential compositional variation scores, and lighter edges don't. This is great, but there is a limitation in that what this approach will do is that it will get features, it will get parts that are most different among many parts. And so if there is a sparse representation where the only thing that's different is maybe between these two, this method will maybe not rank that as high, depending on how significant that separation is. But this method will, in general, sort of reward the parts that are most different among many things. So that's a really important thing. And we'll see that play out later in some of our benchmarking. And so we use this idea in networks where we can compute the node strength, or in this case, we call it the differential compositional variation, or DCV strength. And then from that, we can then apply our constraints. So if we're working with a collaborator and the most parts that they can use are 10, then we can use this information that we now have in this graph and then identify the 10 parts that we think are most predictive. And then from that, we can go in and do a more targeted pairwise log ratio analysis, and we can get a candidate subset that we can use. That's great, but the thing that I note here is that, and I showed earlier, is that this is a supervised approach. And so the question then becomes, how do we do this responsibly? And so we have to be very careful, because we have a lot of noise and we have a lot of dimensions. And so we can get lucky. And we want to make sure that we're doing this in a way that we're not getting lucky because of that. And so what I'll show now is our framework and the way that we sort of have developed it to ensure that we can use this supervised technique and extract robust estimations. And some of the work we've done in our WashU collaboration on the COVID project has really demonstrated the power of this approach, where if we weren't sort of careful up front and sort of setting our expectations for the predictive power, if we sort of overfit and we think we're going to have a model that's going to perfectly predict, and we apply it in real life and it does horribly, that is the absolute worst case. And so this framework really guards against that. And so that's a really important thing I want to say. And so at a very high level, what we're doing in DiCoVar is we're performing sort of a nested set of feature selection. This increases the computational time, but it increases it in a way where we can be more confident in our prediction and what we're estimating the model to be capable of doing in samples that we may not have actually seen. And so I'll just kind of briefly sort of overview this, but if we have a data set where, in this case, we've sort of flipped this matrix sideways. Oh, wow, I'm actually not pointing on the right screen. I do apologize. If we flip this matrix sideways, And so we have our samples here and our parts here. We can split this into K folds. And so this is a really common technique. And so K is determined by the user, which is usually based on the data set size. So we have a lot of samples. We can choose a smaller K if we have, I'm sorry, we can choose a bigger K. If we have fewer samples, we might want to choose a smaller K. And we can use other information to further sort of select these folds and these partitions in a way that's meaningful. So the thing here, we have our outer cross validation loop where we'll use to actually estimate our metric. You'll note here, I put a generic metric term in that really the approach is flexible. We can compute AUC, accuracy, whatever metric you're most interested in, and you can compute that quite flexibly within this framework. But nevertheless, that each partition is analyzed separately. And then so we look at our feature selection and model selection within each fold. And so in my next slide, what we're going to do, I'm going to kind of like walk through more specifically like what happens. So we take that partition, so we can take a single partition, we take what we call the discovery set, we further split that into twofold cross-validation, and then this is where the nesting happens. And so what we're trying to do at this point is figure out what model should we use and what target part. So if your constraint was, I can measure up to 10 parts, then we may test not just 10, but we may test 5, 6, 7, 8, 9 different parts. So we want to find out, given these data, which subset may be most useful given your sort of upper bound on the number of features you want to include. And then we perform, and we have an inner cross validation loop that we apply, and this is the main component here, our targeted multi-level feature selection algorithm. And so from that, we can extract our model that we would want to use, and we can extract a number of parts. The key takeaway here is that the target parts are defined by the user. We can do this also empirically in an untargeted fashion. And the models are, by default, the way that we designed it, either a regression model, which is a ridge regression, or an ensemble model. And so the ensemble models are more complicated, less interpretable modeling set of models that sort of in general performs better for prediction. So if your only goal is to have the most predictive model possible, we would probably just default to the ensemble. But if your goal is to have a predictive model, but you actually want to understand what's happening, then we would probably want to opt for our regression model. And so after we do that, we perform our multi-level feature selection on that. And so what I want to do here is sort of walk through just super quick our pairwise log ratio transformation. So we're taking our discovery data, we're computing, we've seen this previously now at least, our pairwise log ratios. I expanded out to kind of give rise to sort of like the expansion of the dimensions. And then again, this being supervised, we know the group labels, and then we can compute our DCV network, we can compute our target parts, we do our pairwise log ratio at the first level of discovery, and then we train our model. So we've given it the candidate subset. We don't know at this point exactly how well this subset will work. We allow the ridge regression or the average ensemble models to sort of take that subset and then identify and optimize the relationship between the features or that subset and the phenotype of interest, and we can compute our classification metric. So given that, I've kind of given you an idea of how the method works, and what I want to do now is kind of show you some ways, and this is just sort of a snippet of the ways that we benchmark this. We simulated data. We use simulations, a specific type of simulation on the compositional data, but for the purposes of this, we can think of that as a multivariate normal distribution. And so given that, we can take a set of mean vectors in that normal distribution, and we can define sort of arbitrarily or in a random fashion some mean parts, and then we can actually then shift those. And so we can shift the components such that we're going to simulate these data where this percent of the mean parts are actually shifted by this percent. So, and what this represents is the signal density and sparse associations that I was referring to earlier. And so you can think of this as, in the case of 2%, we're saying that if we have 100 features, only 2 of those features are different at 18%. And so we're going to simulate those data. And so this would represent a truly sparse association from a multivariate normal distribution. We use an additive log logistics distribution, so we do this on the simplex. But again, for the purpose of this, we can think of that as a multivariate normal. And so what we're doing in our simulation, we simulate data from a 16S a set of data, and we do this from a whole genome. As I mentioned earlier, they're different. They have different levels of resolution, so they have quite different properties in practice as well. And so that's why we're kind of demonstrating the framework on both types of data. And the takeaway here is primarily that our approach does okay relative to the other approaches. And I should also note, we compared our method against two recently published methods that are specifically compositional. One of which is the Senate log ratio lasso. It's a regularized regression, but it uses a lasso penalty. It's not super important for this talk. It's really identifying the smallest possible subset. And there's a coda lasso, which takes more advantage of sort of this idea of balances. And so it creates sort of a lasso within a sort of a log contrast format. And then we have a SEBL, which is a selection of balances, which is a more balance-driven sort of feature selection technique. All of these are compositional, and so they all recognize the same problem that we're recognizing, but they do it in a different way. And so what we're showing essentially is that in general, for this particular example, those methods perform slightly better than our method for sparse associations. However, as associations become more dense, which is more likely to happen, and you'll see in our next example, more realistic, that when we have more dense associations, we perform significantly better in 16S data, and we perform moderately better in WGS data. However, as the mean shift becomes bigger and bigger, which I'm not showing here simply from the perspective of clarity, is that we do better. So that's the takeaway on synthetic data. But for real data, which is where it's most important, you can see that our approach, which are given by these solid colors, either if we use a pairwise log ratio, additive log ratio signature, or sort of a hybrid signature, which I'm sorry, I apologize, I didn't mention either of these two. We can just think of pairwise in this context. But you can see any of our approaches outperforms the existing data for WGS, which is the more complicated data. And we, in general, outperform the other methods in the 16S data. But this is, most importantly, real data where you'd actually apply this. And so this is where we really do demonstrate the benefit. So if we were developing a diagnostic, and I told you that DiCoVar can predict in this particular case at, well, let's say here at 82% roughly, and then there's another test that may work at 70%. I think in general, if you're trying to predict the disease, in this case, colorectal cancer, you would prefer to have the higher accuracy. And again, because we do this in a robust way, we can be pretty confident that this is a reliable estimate of that predicted performance. So lastly, I want to just kind of walk through a case study where we actually apply these data and apply our technique. In this case study, we were looking at predicting the onset of necrotizing enterocolitis in preterm infants. There was a public study that was published, and because microbiome data is more expansive now, and we're seeing more and more public databases storing and compiling and processing these data, it's quite easy for researchers like myself to access this data and then go in and perform additional analysis. And so here, as I'm citing the source of the original study and actually where we extract the data from. So the study design was such that there were 160 infants were premature, and they sampled the fecal microbiome. And of those, 34 went on to develop this really nasty NEC infection, and some of those, unfortunately, some of those infants died as a result. And so we have control samples. So this is a classic sort of binary classification. And so what we were interested in doing is applying our method to predict, see if we could actually predict with some early warning whether or not an infant was going to go on and develop necrotizing enterocolitis. And so just as an overview of data, when we pre-processed it, we did additional quality checks. We ended up with 144 infants. 120 of those were non-NEC, so controls, and then 24 of those were actually infants that went on to develop this infection. And we had a total of 902 samples. So this creates an interesting problem because this is a longitudinal study. And so we have to be really careful in our cross-validation to account for the fact that this is longitudinal data and we have multiple readings on the same sample. And so we also needed to develop a threshold for when we would expect the model to most likely perform. So like the early warning threshold. And so what we sort of set on was 7 days. And so what we want to do is be able to look at a sample and say with some level of accuracy whether or not in the next 7 or more than the next 7 days, would you develop this infection? So a week from now, or any point after a week from now, would this infant be likely to go on and develop NEC? That would perhaps give enough time for physicians to intervene with antibiotics or something like that. And so we wanted to basically test, given these data, could we do a prediction of that type? And so we also know that there's a severe class imbalance, which creates an additional problem that we have to address. And so with class imbalance, we have to use metrics, or at least a metric that's sort of independent of the imbalance. So we would not use something like accuracy in this setting, because you can guess at random and get 80% roughly at random. So if we built a model and then we said it predicted at 80% accuracy, it'll be a totally useless metric, because you could actually show with simple simulations that you could do that at random, just literally guessing. And so what we do, so ultimately our cross-validation scheme is 20 repeats of 5-fold cross-validation, and we stratify, and so we separate these data by NEC status and infant. And so that way we're making predictions and we're learning and training on sets of infants and predicting on different sets of infants. So we don't want to take a sample from an infant we know is going to develop NEC and then learn patterns that we wouldn't have naturally known to begin with. And ultimately the result when we applied DiCoVar was that we got 67%. We do this in a matched permutation design, and we can actually show that the 67% that we got was actually better than what we would expect at random. So we know not only are we able to predict roughly around 67 or 68%, but we're doing it in a way where we're actually identifying some real pattern in the data. So that's a really important step that's often overlooked in classification studies. And so then the added benefit, we've sort of, again, we've done the machine learning, we've done the prediction, but now the interpretability of the model. How do we figure out what the model learned, what's important? And so the benefit of using our approach is that we get this pairwise log ratio signature. And so what we're saying is that given those data, these are the taxa among the 250 that we actually looked at that were most associated with the prediction. So these are the things that if you look at all of these things, if you're going to develop a diagnostic, these are the things that you want to measure. Most importantly, that these things are relative. So we would never look at any sort of taxa here independently. So the prediction model is such that this network here is the signature, and the interpretation is relative to this network. And so from that, because we use a ridge regression, we're able at this point to then create these, what ultimately is a log contrast, and log contrast has the property of this, our coefficients in our regression model, when we perform some log algebra, essentially will sum to 0. And so, and we can create, and the benefit of using pairwise log ratios with linear models is that you can always create log contrast. And the log contrast provides us with an even better interpretation of the signature, such that in this case, when Staphylococcus and these other taxa were more abundant in this network, you are more likely to be to go on to develop the NEC infection. Whereas when Enterococcus and Pseudomonas, when these things were more abundant, you are more likely to not develop the disease or the infection in the future. So these are what's allowing us to get that early warning system. And the way, and I show this in that this is the scores, but this helps us understand the prediction. So we can't interpret this without this. And so this basically tells us, again, in this sort of equation, which is just a linear model, how to interpret that relative to the actual things that we observe. The other thing here that I thought was really important, and I should note, is that we wanted to then examine, well, when does the model perform best? So is the model performing better in the first 7 days, or is it really 20 days later? And what we find is that the best performance is basically a 10-day warning. So roughly when you're about to develop this thing within the next week or a week and a couple of days, the model you can see performs quite well. Again, that 67% that we quoted was overall, so it includes all these. But if we wanted to sort of take a more granular approach, we can look specifically here and see that the model is actually doing really well when we have enough time. So when there's enough time, there seems to be some change or shift in the microbiome that's giving rise to the onset of this infection. But as we get 24 and 40 days out, we're not as confident, which sort of makes sense in that context. And then the thing that was most impressive, I thought, or most important, is that we trained the model on whether or not you develop NEC. We did not train the model on whether or not you would survive. However, when we actually look at our scores, we notice that the scores that are higher were associated with infants that did not survive. And we see this beyond random. And so again, this isn't an overfit. We didn't train specifically on survival. We're literally just taking our scores for those who developed an infection, and we're actually looking at the distribution of the scores of the survivors versus non-survivors. And so that then means that if I just back up here, when we start to sort of analyze our coefficients, then there could be a set of taxa here that as they become more abundant, they could really be contributing to the ultimate demise of the infants in this case. And so again, this is where we take our DiCoVar approach, we do some prediction, but we then take the interpretability side of it and try to better understand, which again, in traditional machine learning, you just want to predict. Here, we're not just interested in predicting, we have additional constraints, and we're also trying to understand what we're predicting, which is important in the biological sense. So in conclusion, so that basically concludes our sort of presentation for the DiCoVar. So I just want to kind of point out some of the things that are most important is that relative microbiome data are compositional. So if you take away anything, take away that. We introduce our DiCoVar framework, and it's pretty robust. And so, it does a great job for classification, particularly with compositional data sets. I hope we've demonstrated, at least in this talk, and we'll do it in the paper as well, the appropriateness for supervised classification, that we're really careful about how we actually do our supervised feature selection. Our framework is flexible, so if you're an investigator and you're trying to develop a diagnostic, but you have constraints, we can handle that with our framework. And if you're just more, hey, I want the most predictive model possible, we can do targeted analysis as well at the expense of computational time, but we can do it in that fashion as well. And that we have strong predictive capability, but we also have the ability to interpret the model, which is perhaps most important in the biological sense. And in image recognition and other fields, we don't care if the model can, what it learns, if it can pick out a cat versus a dog, but biologically, we really want to understand, well, what's contributing to that prediction. And that's one thing we were really careful in our framework to make sure that we were capable of doing. That said, our package, we have an R package for this as well, that's available here. And so if you're interested in it, we have functions that actually perform all of the steps that I mentioned, and it's fairly reasonably efficient. So that's the conclusion. So I just want to, before I get into my questions, I just want to go through a few things I thought were really important. So I do want to dedicate this defense to my late grandparents who were, I was worried I would get a little choked up with this part, but they were really instrumental in helping me get to this point and keep me on a straight path. There were times where I wasn't always the easiest person to deal with, and without their support and adopting me and my brother at a really young age, I don't think I would have made it to this point. Yeah, I was worried about that. But so my daughters are also, as I said, Madison has been a really, this is really tough, but she's been a really important part to me taking this trajectory and going down a research path, and my daughter Gabby. And I just want to show them that, if you work hard enough, you can actually get things done if you follow your passion and you stay motivated and focused. And so hopefully what I can demonstrate by going through this process to them is that, you can work hard, and it's amazing what you can do and the things you can accomplish. The other thing I would like to acknowledge is my wife, because none of this would be possible without her support. She's literally been beside me the entire time. She has really been as much a part of this as any other person, including myself. We started out in high school, we got married young, and we're still here 20 plus years later, defying the odds of young marriages. And so I just really want to take some time to recognize her and her contribution to me being able to actually undertake this and continue to pursue this research journey. Family has been a really important part. My mother-in-law has been a really strong supporter. She has been really helpful in us making this transition here and making sure we had everything we need to successfully to move from Virginia and get transitioned. My aunt and uncle, Jake and Linda, this is Kathy, that are extremely instrumental, have been excellent sort of role models for me growing up, and really helped me. But have really helped me as an adult. Losing my grandparents young, I didn't really do this, but losing my grandparents young was difficult, and I needed an example, and they really stepped up into that role, and it was really, really important for me. Yeah. And so moving on from that, I really have to acknowledge my co-mentors, day to day at least. Peter and Mike have been an excellent team. They've helped me work through problems. I see things totally different. When I'm working with Mike, we can look at things biologically. When I'm working with Peter, we can look at things mathematically, and I'm able to take those two really unique and strong skill sets and try to merge those in a way that creates this synergy, and hopefully it shows through the work that we've been able to accomplish so far. I have to thank my lab members from the Berks and Coolis lab and the Mucka lab. They've really been really helpful in talking through ideas. Kelly, actually, before I even became a student, she took some time and showed me around the lab when I was considering even making this transition. And she took a few days over the summer and to actually walk through these things. And it was really, really helpful for me to see this life and how it would actually look. And not to underscore any other person here, but I thought I would point that out. And everyone really has been helpful. William and I shared an office, and we talked through a lot of the DiCoVar work. Natalie was a really strong influence, and her dedication and her ability to think through the science was something that rubbed off on me in a really short time, really early on. And again, not taking anything away from anybody here, it's just a few notable things there. Acknowledge our collaborators, the Jeff Henderson lab. I've been fortunate to work with Peter in that lab. Working with Laura and John and Jeff has really been eye-opening, and thinking about metabolomics. They helped me actually think through like why I understand theoretically why ratio is important, but often I get sort of validation from them why biologically, especially in metabolomics, ratios are really important. Working with Elise has been really fun over the last few years. We've been working really hard on an ESIG paper. And I'm glad she included us in that collaboration in the Jaspers lab. And a special thanks to Wesley Burks, who really helped me out at the advice of my daughter's allergy. She was like, "Yeah, you should reach out to Wesley and talk to him about research." And I randomly sent him an email back in 2016, and he actually responded. And it sticks out to me to this day. And he invited me down to see the lab when I worked with Kelly. And it was really instrumental in convincing me that I can actually make this transition, which was difficult because I was working, I was full-time, I had a lot of responsibilities, and then even just questioning whether you have the ability to do this level of work. And then also Lee Anderson, which was one of my former managers while I was working at Newport News. He was really instrumental in helping me continue to stay in school, in the sense that we have a pretty rigorous schedule. And he was really one of the few managers I've mentioned that were flexible, that actually understood that going to school was really important. In some situations, I've had to flex entire days. And again, that's uncommon in that world. And without that level of support and encouragement, I would have never been able to get through the undergraduate, which could have prevented me from getting here to begin with. So I really want to point out his contributions, and we continue to stay in contact throughout this process. And lastly, my funding sources, as Peter mentioned, we had a T32 training grant appointment early on, and the 3-year Howard Hughes Medical Institute Gilligan Fellowship, which has been really helpful for me, allowing me to continue to do this research and funding. And with that, I'll take questions. It's right at 1 hour.
Hello?
Yes, hello?
Hi, Noor Shahir here. Awesome talk, Andrew. It was really impressive. Quick question, and this is probably more of a nitpicky question type thing, but I noticed most of the examples you're using were based on dichotomous outcomes, so like sort of like disease or non-disease. What do you think of your method and other similar methods regarding diagnostics, regarding more continuous variables such like blood pressure and things of that ilk? Like, do you have any insight or things like that?
That's an interesting question and a really interesting point. We actually didn't spend much time looking at continuous variables. So everything here was more classification, binary multi-classification types of problems. So I haven't actually spent much time looking at that. And I think it will be similar in a regression sort of way. But for sure, I think that is an interesting area, an important area where you want to do some association analysis. But we haven't actually spent much time actually looking at continuous outcomes.
I have a question.
Sure.
Hey, Andrew, congrats.
Thanks, Ian.
It's really fun to watch. I always like watching you talk about this stuff. Yeah, it's just kind of a general microbiome question. So all your stuff is working with compositional data, right? Where you have relative abundances of things because of the limitation of the experimental technique. You can't really interpret the absolute magnitudes of the, yes, each group. I'm wondering, and you obviously see a lot of like, you can make a lot of detections using just the relative abundances, but do you know how much more power you would gain from being able to interpret the magnitudes?
There have been some approaches that have looked at that. Generally, in my experience, I actually did that sort of naively up front. And just from my experience, I didn't see improvements. In fact, it got more difficult because while the example I showed things were at a sort of like a reasonable level of variation, in reality, you can get orders of magnitude differences where one sample may have 10 to the 8th and another sample may have 10 to the 3rd. And then that sheer order of magnitude difference can skew almost any prediction model. And so in general, you really would have to normalize unless you had sort of equal sort of variation within that count space.
Cool, that's really interesting, thanks.
Any further questions for Andrew? All right. Well, why don't we, I see a lot of well wishes and congratulations coming through the chat. Everyone's free to include some more there. Wish Andrew well, send him a reaction by Zoom, and then I'd like to ask everyone to be dismissed here by 10:40, but ask the committee to stick around for the private exam. It was really great to see friends and family. I was telling Andrew just before his really nice presentation, one of the silver linings, I guess, to doing this by Zoom is that folks who are far away can attend. So it was great to see all of those supporters there. So well done, Andrew. Thanks again. And let's now dismiss everyone for the private defense. Thank you.
Thanks everyone for coming. Really appreciate it.